Hello, everyone. The purpose of this lecture is to supplement the content of the Genetics of Pediatric Brain Tumors chapter in Humans and Wind Neurological Surgery. My name is Kyle Yarushka, and I am a neurosurgery resident at the University of Toronto, and I'll be presenting on behalf of my co-authors Sachin Kumar and Dr. Michael Taylor. This lecture will be organized similar to the textbook chapter. First, we'll discuss the genetics of gliomas, first high grade and then low grade. I'll subsequently outline embryonal tumors, including medulloblastoma, ATRT, ETMR, and other entities. We'll next discuss ependymal tumors, germ cell tumors of the central nervous system, craniopharyngioma, choroid plexus tumors, pineoblastoma, hemangioblastoma, and vestibular schwannoma. By the end of this lecture, I hope that you will have achieved an understanding of the key genetic alterations and molecular pathways that drive tumorigenesis in each of these entities and understand how newly defined molecular entities are starting to change clinical practice. Pediatric gliomas can broadly be divided into high-grade gliomas and low-grade gliomas. The discussion of high-grade gliomas will be organized by anatomical compartment, as the genetic alterations correlate strongly with anatomical region. The discussion of low-grade gliomas will generally be organized according to known histological entities and can be subclassified as glial or mixed glial neuronal entities. High-grade gliomas in children are clinically aggressive and difficult to treat. The genetic landscape of these tumors is quite different from high-grade gliomas in adults. Perhaps the single most defining feature of pediatric high-grade gliomas is the presence of recurrent mutations in genes encoding replication-independent histone 3 variants. These mutations are the defining features of almost all DIPG, the majority of non-brainstem midline gliomas, and approximately one-third of hemispheric high-grade gliomas. The remaining group of hemispheric tumors is a collection of heterogeneous tumors, and recent research has attempted to better classify these entities based on methylation signatures. We will approach this section in a top-down approach, starting with hemispheric tumors, followed by non-brainstem midline tumors, and finally DIPG. Of note, a small number of pediatric high-grade gliomas can occur in the setting of cancer predisposition syndromes, such as Li-Fraumeni syndrome, which is caused by TP53 mutations, and biallelic mismatch repair deficiency, but we will not discuss this further in this section. As mentioned, histone H3 variants are at the core of pediatric high-grade glioma biology, and hemispheric tumors are no exception. The H3.3 variant is very similar to the canonical histone H3 and commonly replaces the canonical H3 at transcriptionally active genes and promoter regions. It also appears to accumulate at silent loci in the pericentric heterochromatin and telomeres. H3.3 G34R/V primarily occurs in teenagers and has a worse prognosis compared with H3.3 or H3.1 wild-type tumors. However, outcomes are better than cases with H3.1 and H3.3 K27M mutations that are seen in midline high-grade gliomas. The H3.3 G34R/V variant occurs with TP53 mutations in over half of cases. The most highly recurrent event in this group are mutations of ATRX and DAX. Together, these mutations result in alternate lengthening of telomeres, which is a mechanism common to cancers that allows maintenance of telomere length without telomerase. An additional proposed mechanism of tumorigenesis is that this H3.3 mutation results in chromatin remodeling, causing a distinct transcriptional program with genes involved in stem cell development, cell fate, and self-renewal. MIC-N expression is also upregulated. Among pediatric high-grade gliomas, this and IDH1 mutant tumors are the only subgroup that demonstrate MGMT promoter methylation. IDH1 mutations are relatively rare in pediatric high-grade glioma, accounting for only 6% of cases. IDH1 mutant high-grade gliomas occur in supratentorial hemispheric locations. They occur in teenagers and into adulthood. Thus, children likely represent the early end of the age spectrum of this disease. Outcomes are better than other pediatric high-grade gliomas, as outlined in this survival curve in the bottom right. IDH1 mutations are considered to be an early driver event in gliomagenesis, priming neural progenitor cells for additional transforming events such as TP53 and ATRX mutations. Additional alterations such as RTK pathway activation or PTEN mutations are thought to cause transformation into high-grade glioma. These tumors feature a glioma CpG island methylator phenotype, which has been associated with silencing of mesenchymal genes, which is a suggested reason as to why this subgroup tends to be less clinically aggressive. To classify histone 3 variant and IDH1 wild-type tumors, a study by McKay and colleagues proposed 3 methylation subgroups, namely wild-type A, wild-type B, and wild-type C. The wild-type A subgroup was composed primarily of PXA-like and low-grade glioma-like entities. The PXA-like group is named for its molecular similarities to the low-grade entity pleomorphic xanthoastrocytoma. These tumors primarily occur in hemispheric locations and at a median age of 11 years old. Like PXAs, which will be discussed later in the low-grade glioma section, this group has a high rate of BRAF V600E mutations, which co-occur with CDKNA2A and B loss. 1q loss is a poor prognostic factor in this subgroup. Overall, these tumors appear to have a relatively intermediate prognosis among pediatric high-grade gliomas. Interestingly, given the genetic drivers of these tumors, targeted therapy with MEK or BRAF inhibitors has been shown to have promising results in early clinical trials. A subset of high-grade gliomas have methylation patterns that are similar to low-grade glioma entities. Children with these tumors have a younger median age at diagnosis and the best overall survival. BRAF truncations are a common recurrent event. Additionally, there appears to be a distinct cluster within this subset that are driven by NF1 mutations. Finally, a unique set of these tumors arise in infants that are 12 months of age and younger, and these are driven by NTRK1-3 fusions. These cases have excellent outcomes and drive the good overall outcomes seen in this group. As you can see in the survival curve on the bottom right, the children less than 1 year have quite good survival, whereas older children, whether they're 1 to 3 years old or older than that, have quite poor survival. In recent years, attempts have been made to better understand pediatric high-grade gliomas that are not caused by IDH1 or histone 3 variant mutations. 2 research groups have proposed subgroups, and there appears to be some overlap, with the McKay wild-type C subgroup resembling the PGBM-RTK1 subgroup, and survival curves appear to be similar. The wild-type B subgroup has drivers that are similar to the PGBM-RTK2 subgroup and the PGBM-MYC-N subgroup, suggesting that these 2 classification systems are identifying biologically similar subgroups. As described in previous slides, the McKay wild-type A subgroup is composed of low-grade glioma-like and PXA-like tumors. Wild-type B tumors are driven by amplifications of EGFR, MYC-N, and CDK6. Wild-type C tumors are driven by PDGFR-alpha and MET amplifications. Korshunov and colleagues have subgroups that include a group driven by MYC-N and ID2 amplifications that have a high rate of TP53 mutations and poor survival. The RTK1 subgroup is driven by platelet-derived growth factor receptor alpha amplification, and the RTK2 group has enrichment of EGFR amplification and CDKNA2A and B deletion. Moving on from genetic entities that primarily cause hemispheric tumors, histone 3.3 variant K27M mutations account for the majority of both non-brainstem midline high-grade gliomas and DIPG, as pictured in the lower right figure, with larger light green circles representing relative high abundance of these tumors in various anatomical locations. They are associated with an older age of onset than H3.1K27M mutant tumors and have less than 5% of patients surviving at 2 years. The H3.3K27M mutation results in global loss of H3K27 trimethylation by interfering with the polycomb repressor complex 2. Non-brainstem H3.3K27M tumors harbor FGFR1 gene variants, NF1 mutations, and CDK4 amplifications, and less frequently have TP53 mutations. In contrast, H3.3K27M DIPG frequently contains co-occurring mutations of PDGFR-alpha and TP53. H3.1K27M mutations account for most of the non-H3.3K27M mutant DIPGs. These mutations are rarely observed in other tumors. Age at diagnosis is typically between ages of 4 and 6. These tumors are associated with very poor survival, but it is marginally better than H3.3K27M tumors. Molecularly, they are also associated with a global H3K27 hypomethylation pattern. Nearly all of these tumors contain ACVR1 mutations, resulting in SMAD phosphorylation and BMP and TGF-beta signaling activation. Now we'll move on from high-grade glioma to low-grade glioma. Before we begin discussing specific low-grade glioma histological entities, I'd like to highlight the importance of the MAPK signaling pathway in pediatric low-grade glioma biology. In some respects, this can be considered a single pathway disease, as nearly all low-grade glioma entities have activation of MAPK signaling through alterations noted in almost every component of this pathway, as seen in the figure on the left. As such, targeted therapy for low-grade glioma has undergone rapid clinical translation, and agents such as MEK and BRAF inhibitors have shown promising responses in early-phase clinical trials. Low-grade gliomas can broadly be subclassified histologically into glial tumors, containing most astrocytic and oligodendroglial tumors, and neuronal or mixed glioneuronal entities. In children, these tumors generally have excellent prognosis with high rates of long-term survival, although tumor progression can occur, especially in higher-risk entities. Pilocytic astrocytomas are the most common low-grade glioma diagnosis in children. Pilocytic astrocytomas can occur in the setting of neurofibromatosis type 1, most often in the optic pathway, caused by a mutation in the NF1 gene. Normally, this gene product inhibits RAF and prevents MAPK pathway activation. Patients with Noonan syndrome, another RASopathy caused by PTP and 11 mutations, also develop pilocytic astrocytomas. Sporadic pilocytic astrocytomas most commonly arise in the cerebellum, and the genetic driver event is BRAF fusions. Its most common fusion partner is KIAA1549. BRAF V600E mutations are also observed. Diffuse astrocytomas in children have genetic alterations that vary by anatomic compartment. Hemispheric diffuse astrocytomas often contain amplifications or rearrangements of MYB and MYB ligand 1. When they arise in midline structures, BRAF fusions, BRAF V600E mutations, and NF1 mutations predominate. Pleomorphic xanthoastrocytomas, or PXAs, are grade 2 tumors, but have a more aggressive clinical behavior. Like the PXA-like high-grade gliomas described previously, these tumors have a very high frequency of BRAF V600E mutations, which occur with a loss of 9p21, the locus of CDKN2A and B. Oligodendrogliomas are rare entities in children, and unlike adult oligodendrogliomas, these usually do not have IDH1 mutation and 1p19q codeletion. Instead, MAPK alterations underlying pediatric oligodendrogliomas via FGFR1 tyrosine kinase domain duplications, FGFR1 mutation, BRAF V600E mutations, and FGFR1 TAC1 fusions. Subependymal giant cell astrocytoma, or SAGA, also known as subependymal giant cell tumor, or SGCT, occur in patients with tuberous sclerosis complex. This is caused by germline mutations of TSC1 or TSC2, which are components of the PI3K mTOR pathway. Randomized trials have shown that treatment with mTOR inhibitors such as everolimus result in tumor regression or prevent further tumor growth in nearly all patients. The seizure frequency in these patients is also reduced with this treatment. Of note, treatment withdrawal tends to result in relapse, meaning that these patients require ongoing therapy. Ganglioglioma is a grade 1 tumor that typically arises in the temporal lobe, causing epilepsy. BRAF V600E mutations cause approximately 50% of the cases, and BRAF fusions are seen in a significant minority of the remaining of these cases. Dysembryoplastic neuroepithelial tumor, or DNET, are the most commonly caused by alterations of FGFR1, including tyrosine kinase duplications, mutations, and fusions. 5 to 10% of cases are caused by BRAF V600E mutations and other tyrosine kinase alterations. Desmoplastic infantile astrocytoma and gangliogliomas are rare tumors that occur in very young patients. MET gain appears to be a common driver, as well as BRAF V600E mutations, EGFR amplifications, and MIGAN amplifications. A collection of rare glioneuronal tumors have also been shown to have highly recurrent genetic events. Angiocentric gliomas contain MIB and MIB ligand 1 fusions, as seen in diffuse astrocytoma. Choroid gliomas of the third ventricle are driven by PRKCA mutations, while papillary glioneuronal tumors contain fusions of PRKCA to SLC44A1. Rosette-forming glioneuronal tumors have high rates of FGFR1 mutations, which often occur with PEIK3CA and NF1 mutations. Finally, polymorphous low-grade neuroepithelial tumor of the young, or PLENTY, has MAPK pathway activation via mutually exclusive FGFR2-3 fusions or BRAF V600E mutations. We will now move on from gliomas and discuss embryonal tumors. The molecular era has truly refined the way we classify these tumors. While many of these entities used to be classified as primitive neuroectodermal tumors, this term was removed from the 2016 WHO classification of CNS tumors. Moreover, new distinct entities have emerged, and subgroups of well-known entities have granted us new clinical and biological insights for these diseases, which generally have poor outcomes and are in need of improved therapies. We'll discuss these entities individually in more detail in the coming slides. We'll begin by discussing medulloblastoma. Medulloblastoma is now recognized to consist of 4 molecular subgroups: Wnt, sonic hedgehog group 3, and group 4, which each have important clinical and molecular features. An overview of the subgroups is provided here, and we'll discuss each subgroup in subsequent slides. Since this initial subgroup definition, subgroups have proposed further molecular subtypes of medulloblastoma, which will also be touched upon briefly in this section. However, it is important to recognize that this is an evolving area of research. The Wnt subgroup is the rarest subgroup, accounting for only 10% of cases. It is absent in infants but occurs in all other age groups. It is associated with excellent prognosis with overall survival exceeding 95%. Progenitor cells in the lower rhombic lip are the suspected cell of origin in the subgroup, which may explain why this subgroup often exhibits involvement with the cerebellar peduncles and brainstem. Radiographically, this results in a tumor location centered in the cerebellar pontine angle in some cases, while the remaining majority of Wnt tumors arise in the midline. Another clinical feature of these tumors is that they have a leaky blood-brain barrier, which has been proposed as a reason why these tumors are more sensitive to treatment and why they often have a hemorrhagic component. Molecularly, these tumors are driven by aberrant activation of the canonical Wnt signaling pathway, pictured on the bottom right. Normally, this pathway is activated by a Wnt protein binding to frizzled, which sends a signal to disheveled, which subsequently deactivates a destruction complex that, in the absence of Wnt signaling, targets beta-catenin for degradation. So, when Wnt signaling is activated, beta-catenin accumulates in the cytoplasm and translocates to the nucleus and activates transcription of target genes that cause cell proliferation and migration. Common alterations in this pathway include activating CTNNB1 mutations in the third exon. These mutations result in the activation of the Wnt pathway without the Wnt protein. This aforementioned gene encodes beta-catenin. This mutation frequently co-occurs with monosomy 6 and is the only recurrent cytogenetic change seen in this subgroup. The next most common Wnt pathway alteration is APC mutation, both as a germline mutation or sporadically. Finally, this subgroup has the second highest overall mutational burden, and commonly altered genes are listed in the figure in the bottom right. 2 subtypes have been proposed, alpha and beta. The alpha subgroup occurs in somewhat younger patients, and nearly all of the patients in this group had monosomy 6. In comparison, the beta subgroup was slightly older, and less than 50% of cases had monosomy 6. The sonic hedgehog group is the second most common subgroup. It is quite heterogeneous clinically, and in some respects molecularly as well. It occurs across all age groups, but is most common in infants and children. There is a balanced gender ratio. All histological subtypes are represented, but desmoplastic and MBEN are most frequently observed in sonic hedgehog tumors compared to other subgroups. Recurrences typically occur locally rather than metastatically. Tumors can occur in the cerebellar hemispheres or in a midline vermian location. Older age is associated with tumors in a cerebellar hemisphere location, while younger patients have midline tumors. This tumor is driven by sonic hedgehog pathway activation, as the name suggests. Normally, sonic hedgehog inactivates patch 1, resulting in loss of inhibition of SMO. This leads to signaling cascades through SUFU or MIC-N, resulting in downstream expression of target genes. Multiple sonic hedgehog pathway genes are altered in sonic hedgehog medulloblastoma cases, including loss-of-function mutations or deletions in patch 1 and SUFU, activating mutations in SMO, and amplifications of MIC-N and GLI1 or GLI2. MIC-N amplification has been associated with poor prognosis, along with mutations in TP53, which results in genomic instability. TERT promoter mutations are also frequent, especially in the adult population. Cytogenetic events such as 9q loss, which contains patch 1 and ELP1, 10q loss containing SUFU, or 17p loss containing TP53 are common cytogenetic events that often cause loss of heterozygosity of the aforementioned genes. Subtypes of medulloblastoma have been proposed to help better understand the clinical and molecular heterogeneity within medulloblastoma subgroups. The goal of these further refined subtypes is to provide tools to improve prognostication and therefore affect treatment decisions for clinicians, and to provide researchers with the ability to identify targeted treatments that might be effective within certain subtypes. The proposed subtypes of sonic hedgehog medulloblastoma arguably have the most intuitive clinical relevance, and there is quite strong overlap between the subtypes proposed by Cavalli, Kuhl, Schwalb, and Robinson. The subtypes proposed by Cavalli, namely alpha, beta, gamma, and delta, arguably most holistically capture the entities proposed by the other groups, and I will discuss that system further here. Sonic hedgehog alpha occurs in children and teenagers. This is a higher risk group within the sonic hedgehog subtypes with 5-year survival of 70%. LCA histology occurs in almost 25% of cases, while this is rare in other subtypes. Among the molecular changes outlined in the table on the left, TP53 mutations, which occur in approximately 1/3 of tumors in this subtype, are of particular clinical relevance, since they are associated with survival rates of just over 25%, as you can see in the survival curve on the bottom. Recently, ELP1 germline mutations in sonic hedgehog medulloblastoma were described, and these almost exclusively occur in sonic hedgehog alpha tumors. These mutations are mutually exclusive with TP53 mutations and are associated with over 90% 5-year overall survival, suggesting that there may be additional useful stratification within sonic hedgehog alpha tumors. The beta and gamma subtypes together account for the majority of cases in infants and young children. Clinically, sonic hedgehog beta has poor prognosis and the highest rate of metastasis. Broad chromosomal gains are common, and P10 loss resulting in PI3K pathway activation is enriched in this subtype. Sonic hedgehog gamma is associated with excellent clinical outcomes and contains most of the MBEN histology tumors, which are also associated with good outcomes. Of note, survival in this group is good regardless of MBEN subtype, suggesting that this subtype system may provide a better way of identifying young sonic hedgehog patients with good prognosis compared to histology alone. Cytogenetic changes are rare in the sonic hedgehog gamma subtype. Finally, the delta subtype is essentially an adult disease and survival is excellent. Molecular features include TERT promoter mutations in over 90% of cases and a high overall mutational burden. Recently, mutations in the non-protein coding U1 spliceosomal RNA family were identified in 97% of sonic hedgehog delta cases. These RNAs are responsible for the recognition of splicing sites in pre-messenger RNA, and the mutation results in alternative splicing of multiple genes, including sonic hedgehog pathway genes. These alternatively spliced mRNAs result in protein variants that may make these tumors particularly susceptible to immunotherapy. Group 3 medulloblastoma occurs primarily in infants and young children and becomes rare among adolescents and adults. It is twice as common in males, and LCA histology is most common in this subgroup compared to other subgroups, as depicted by the relatively large orange bar in the histology figure in the middle on the right. This group has high rates of metastasis and the worst survival of all subgroups. These tumors almost always arise in the midline and usually have strong contrast enhancement. Metastasis in this subgroup often display a laminar or sugar coating pattern. The most common recurrent events are MYC amplification, which is associated with very poor prognosis, and GFI1 or GFI1B overexpression via an enhancer hijacking event. Recurrent mutations are quite rare. Instead, epigenetic mechanisms are likely key in group 3 tumorigenesis. Less consensus exists in group 3 and group 4 subtype definitions. In fact, one proposed group 3 and 4 methylation subtype system contains subtypes that overlap between these two subgroups. Cavalli et al. Proposed 3 subtypes of group 3 medulloblastoma, namely alpha, beta, and gamma. Group 3 alpha contains 60% of group 3 infants and has a high rate of metastasis, but is associated with the best prognosis. Group 3 beta has the oldest median age, 7.5 years, and enrichment of GFI1 or GFI1B overexpression. Group 3 gamma is defined by enrichment for MYC gainer amplification and is associated with a worse prognosis, with 5-year overall survival of approximately 41.9%, even when controlling for MYC amplification status. Group 4 medulloblastoma is the most common subgroup and is 3 times more common in males. It is generally a tumor of childhood and adolescence and carries an intermediate prognosis. These tumors are also characteristically midline tumors. However, 60 to 80% demonstrate scant or absent contrast enhancement, which is a defining feature of this subgroup's imaging. The most highly recurrent genetic event is a tandem duplication of SYNCAP, resulting in upregulation of PRDM6 via enhancer hijacking. A number of different genes involved in histone modifications are recurrently mutated, suggesting epigenetic dysregulation may be central to group 4 biology. Isochromosome 17q is present in 80% of cases, and the cytogenetic landscape is often highly irregular. Cavalli et al. Proposed 3 subtypes of group 4 tumors. These subtypes don't result in very obvious clinical phenotypes. There may be a trend towards survival difference between these subtypes, but it is not statistically significant. The distinguishing molecular features are MYC-N amplification for the alpha subtype, SYNCAP duplication and PRDM6 upregulation for the beta subtype, and enrichment of CDK6 amplification in the gamma subtype. Numerous genetic predisposition syndromes causing medulloblastoma have been reported with varying penetrance, and many of them are listed in the figure in the top right. These vary in frequency between subgroups, with up to 20% of sonic hedgehog tumors being caused by genetic predisposition syndrome in a study of 1,000 patients. With the recent report of ELP1 mutations causing up to 14% of sonic hedgehog medulloblastoma, this estimate may be as high as 40%. Approximately 6% of Wnt tumors occur in patients with familial adenomatous polyposis syndrome, while germline mutations are rare in group 3 and group 4 patients. The most common syndromes and mutations that cause the highest proportion of cases are ELP1 mutations resulting in sonic hedgehog medulloblastoma, Gorlin syndrome, which is caused by PATCH1 and SUFU mutations and results in sonic hedgehog tumors, Li-Fraumeni syndrome, which is caused by TP53 mutations and results in sonic hedgehog tumors, and mutations in the APC gene, resulting in Wnt medulloblastoma. Given their embryonal origins, a lot of researchers have attempted to identify the cell of origin for medulloblastoma. Clinically, this likely impacts the difference in anatomical location of the various subgroups, which will be discussed further later. Additionally, it suggests that these cells may undergo transformation events early in embryogenesis. Wnt medulloblastoma likely arises from lower rhombic lip progenitors in the developing brainstem. Sonic hedgehog medulloblastoma arises from granular cell precursors. Group 3 medulloblastoma arises from early progenitor nestin-positive stem cells.
and group 4 medulloblastoma likely arises from unipolar brush cells or glutamatergic cerebellar nuclei, or both. The molecular subgroups may have surgical implications as well. Subtotal resection has long been considered to predict worse progression-free survival, as shown in the figure closest to the left. However, in a retrospective multivariable analysis, there is only a benefit of gross total resection over subtotal resection for group 4 patients, but not the other subgroups. Thus, significant residual tumor in a group 4 patient may warrant consideration of repeat resection if technically feasible, while residual disease may not have a strong prognostic value in the other subgroups. However, the retrospective nature of this study, combined with the fact that tumors with subtotal resection have typically received more aggressive therapy, limits these conclusions and warrants further study. Atypical teratoid rhabdoid tumor, or ATRT, are the most common tumors in infants. These are clinically aggressive tumors that are very invasive and often metastatic. The prognosis is very poor. The hallmark genetic feature is biallelic loss of SMARCB1. SMARCB1 encodes the HSNF5/BAF47/INI1 protein, which is a core subunit of the SWI/SNF chromatin remodeling complex, implicating epigenetic dysregulation in ATRT tumorigenesis. Other mutations or cytogenetic changes are rare, thus the epigenetic changes drive ATRTs. EZH2 inhibitors have been proposed as a potential therapy. Two research groups have independently identified 3 groups of ATRT, Group 1, Group 2A, and Group 2B. Group 1 tumors occur primarily in supratentorial locations. Molecularly, they are characterized by focal alterations in SMARCB1, neurogenic molecular profiles featuring increased Notch and sonic hedgehog pathway signaling, and are hypermethylated. Group 2A tumors typically arise infratentorially and have either focal or broad SMARCB1 alterations. This subgroup has a hypomethylated genome and overexpresses melanosomal genes, BMP pathway genes, and platelet-derived growth factor receptor beta. Finally, Group 2B occur in the broadest age distribution and arise throughout the CNS. Approximately half the cases occur supratentorially, while the remaining cases are divided equally between infratentorial and spinal locations. These tumors typically have broad losses of chromosome 22q encompassing SMARCB1 and have a hypermethylated genome. Key transcriptional alterations including upregulation of MYC and HOX A, B, and C. Similar to Group 2A, Group 2B tumors have upregulation of BMP pathway genes and platelet-derived growth factor receptor beta. Embryonal tumor with multilayer rosettes is a relatively new diagnostic entity. It typically occurs in children under 2. The underlying biology is quite fascinating. These tumors are driven by a fusion event of the promoter of TTYH1 to C19MC, which is a microRNA cluster that is normally exclusively expressed in the fetal brain during embryogenesis. This microRNA cluster is also found only in primates, with a potential role in the evolutionary development of the primate nervous system. This fusion event causes ongoing expression of C19MC, which encodes MIR519A, which inhibits RBL2. RBL2 normally inhibits the de novo DNA methyltransferase gene DNMT3B. DNMT3B is active during embryogenesis, induces pluripotent self-renewal, and results in a unique methylation profile. This biology results in the tumor having histological similarities to components of the developing neural tube. Since DNMT3B is normally inactive outside of embryogenesis, it is a promising therapeutic target. As mentioned, PNET is no longer a histopathological entity under the new edition of the WHO classification system. These tumors were notoriously hard to differentiate based on traditional histopathological features. Classification based on methylation profiles of large cohorts of tumors that were previously diagnosed as PNETs have identified rare but unique molecular entities. CNS neuroblastoma with FOXR2 activation activates FOXR2 by various fusion and deletion events. This subgroup was enriched for CNS neuroblastoma and CNS ganglioneuroblastoma entities. CNS Ewing sarcoma family tumor with CIC alteration is defined by CIC alterations, including CIC NUT-M1 fusions and CIC frameshift deletions. It results in upregulation of the ETS transcription factor family. CNS high-grade neuroepithelial tumor with MN1 alteration contains MN1 fusions. Finally, CNS high-grade neuroepithelial tumor with B-core alteration contains tandem duplication of B-core and has upregulation of the Wnt signaling pathway. Pediatric ependymal tumors can broadly be classified by anatomical location. Within each of these locations, there are additional subgroups with varying clinical and genetic characteristics. Of note, subependymoma may arise in each of these compartments and can be considered an additional subgroup. However, these tumors occur almost exclusively in adults. Complete surgical resection significantly improves outcomes in most of these entities. The supratentorial region is the second most common compartment that ependymomas arise in. There are two major molecular subgroups, ST rela, which is more common, and ST yap. ST rela has a median age of onset of 8 years old and is more common in males. Molecularly, it is defined by a major shattering event on chromosome 11, resulting in a C11orf95 rel-A fusion. The functional role of C11orf95 is unknown, but rel-A encodes a core functional component of the NF-kappa B signaling pathway. Common events occurring with this fusion are CDKN2A homozygous deletion and chromosome 9p loss. Prognosis is good in the case of complete resection, but poor with residual tumor. Thus, attempt at repeat resection should be considered. Radiation is often used as an adjunct, but observation is also considered if there's no residual. ST-YAP tumors occur in younger age groups and clinically have very good prognosis if completely resected. These tumors contain fusions of YAP1 to MAM-LZ1 or FAM118B. Posterior fossa tumors account for nearly 2/3 of pediatric cases, and there are two major subgroups, PFA and PFB. These are histologically indistinguishable, but have very different clinical outcomes. Of note, chemotherapy does not have benefit in the treatment of ependymoma. PFAs occur in infants and children with a median age of onset of 3 and have a 5-year overall survival rate of 68%. They occur much more commonly in males. PFA tumors possess a balanced genome with very few recurrent somatic mutations, but global H3K27 hypomethylation, implicating epigenetics at the core of PFA biology. This epigenetic signature has roots in embryonic development where PFAs arise in a hypoxic environment that drives metabolic programs resulting in H3K27 hypomethylation. The most common mutation, occurring in less than 10% of cases, is CXORF67 or EZHIP. This gene is overexpressed in all of PFAs. Rare H3.3K27M mutations have also been reported. Patients who require radiation and repeat resection should be considered in the case of residual tumor, since this appears to affect prognosis. 1Q gain is associated with very poor outcomes in this group. PFB tumors occur in adolescents and adults and have more promising outcomes with gross total resection and adjuvant radiation. PFBs are genomically unstable with multiple arm-level copy number alterations. 13q loss is associated with poor prognosis in this subgroup. Complete surgical resection is the strongest predictor of outcome in this subgroup, and recent trials suggest observation without radiotherapy can be considered for tumors without 13q loss. Spinal ependymomas typically occur in adults. Spinal ependymoma features chromosomal instability, and chromosome 22, encoding NF2, loss is common. Myxopapillary ependymoma also has a high degree of chromosomal instability, with numerous frequently observed gains and losses. Histologically, CNS GCTs are classified as germinomas or non-germinomatous germ cell tumors, or NGGCTs. NGGCTs include embryonal carcinomas, endodermal sinus tumors or yolk sac tumors, choriocarcinomas, and teratomas, including immature teratoma, mature teratoma, and teratoma with malignant transformation. Approximately 1/4 of NGGCTs are mixed, containing more than one histological component. These germ cell tumors are 3 to 4 times more common in males and far more common in Asian countries. MAPK signaling pathway alterations are frequently observed in this type of tumor. KIT mutations are the most common but occur only in germinomas. RAS gene family alterations are the next most frequently observed. Several other MAPK pathway genes are also mutated in some cases, notably CBL, which normally targets receptor tyrosine kinases such as KIT for ubiquitination and degradation. Another commonly targeted signaling pathway is the PI3K pathway. There are mutations in these pathway genes in 12% of cases, and AKT1 amplifications are present in 20%. Chromosomal instability is a hallmark of germ cell tumors, although this is far more frequent in germinomas. In fact, approximately 40% of NGGCTs have a balanced genome. Germinomas and NGGCTs also have characteristic chromosomal gains and losses, which are outlined in the textbook. The epigenetic landscape differs between germinomas and NGGCTs, with a hypomethylation signature in germinomas and hypermethylation in NGGCTs. In the last part of this lecture, we will discuss several other pediatric brain tumors, including craniopharyngioma, choroid plexus carcinoma, hemangioblastoma, and vestibular schwannoma. Craniopharyngioma contains two distinct subtypes that have been shown to have distinct molecular alterations. Adamantinomatous craniopharyngioma has a bimodal age distribution and occurs in childhood between the ages of 5 and 15 and older adults. These tumors are driven by Wnt signaling pathway activation, and 96% of these tumors have been shown to have CTNNB1 mutations, the gene that encodes beta-catenin. Papillary cholangiocarcinoma is a primarily adult tumor. Over 95% of these tumors have BRAF V600E mutations. Targeted therapy with BRAF and MEK inhibition has shown promising results in early clinical trials. Choroid plexus tumors include choroid plexus papilloma, which is a grade 1 tumor, atypical choroid plexus papilloma, which is a grade 2 tumor, and choroid plexus carcinoma, which is a grade 3 tumor. Choroid plexus carcinoma are a hallmark tumor of Li-Fraumeni syndrome, which is caused by TP53 mutations. Somatic mutations in TP53 are observed in 50 to 60% of CPCs and are associated with increased genomic stability, tumor aggressiveness, and poor survival. Almost all choroid plexus tumors contain chromosomal imbalances, and the chromosomal gains and losses observed in CPCs and CPPs are different. Of note, chromosome 9p gain or 10q loss portends better prognosis in choroid plexus carcinoma. Several groups have used methylation signatures of choroid plexus tumors to identify subgroups. Of note, the clustering method used by Thomas et al. Identified some choroid plexus papilloma and atypical choroid plexus papillomas that co-cluster with choroid plexus carcinomas that appear to have a higher risk of progression. This is an evolving area of research, but if reproducible, may become a useful diagnostic tool to identify high-risk, low-grade tumors. Pineal parenchymal tumors include pineocytoma, pineoblastoma, pineal parenchymal tumor of intermediate differentiation, and papillary tumor of the pineal region. Pineoblastoma occurs in pediatrics, while the other entities are rarely diagnosed in children. Pineoblastoma can occur in the setting of germline RB1 mutations, occasionally co-occurring with bifocal retinoblastoma, a condition termed trilateral retinoblastoma. Other common driver events are microRNA processing gene mutations, including germline dicer1 mutations and sporadic DROSHA and DGCR8 mutations. Recent integrated molecular profiling by several research groups have proposed up to 5 subgroups of pineoblastoma. One such subgroup classification system is provided on this slide. While consensus subgroup definitions are not yet defined, commonalities exist between these classification systems. There appear to be 2 high-risk subgroups that occur in infants and have very poor overall survival. These tend to feature RB1 inactivation combined with gain of MIR-1792 microRNA cluster or mic amplification. 2 other subgroups with good clinical outcomes contain loss of function alterations of miRNA biogenesis genes, including dicer1, DROSHA, or DGCR8. These tumors occur in older children and adolescents. Finally, a subgroup that appears to have molecular overlap with pineal parenchymal tumor of intermediate differentiation, which contains KBT-BD4 mutations occurs in adolescents and adult patients. Medulloblastoma can occur sporadically and in von Hippel-Lindau disease. While across all age groups almost 75% of hemangioblastomas are sporadic, VHL disease causes hemangioblastoma formation at earlier ages and thus accounts for most pediatric cases. The VHL hypoxia-inducible factor, or HIF, signaling axis is normally activated in hypoxic conditions, upregulating target genes such as VEGF, GLUT1, and EPO. In VHL patients, loss of heterozygosity events of chromosome 3p, the locus of VHL, is required to initiate tumorigenesis. Some sporadic cases contain HIF2A alterations, which is an alternate mechanism of activation of HIF signaling. Therapies targeting EGFR, or the PI3K pathway, have been proposed to treat these tumors, which often arise in surgically high-risk areas such as the brainstem and spinal cord. Vestibular schwannomas develop from Schwann cells in the superior or inferior branch of the 8th cranial nerve. In children, they almost always occur in the setting of neurofibromatosis type 2, a neurocutaneous syndrome characterized by cafe au lait skin markings and the development of benign tumors. 98% of these patients eventually develop bilateral vestibular schwannomas. Thus, this is considered pathognomonic for NF2. This is an autosomal dominant condition caused by mutations in the NF2 gene, which encodes a protein called merlin. Merlin is a tumor suppressor gene with roles in cell motility, cell adhesion, and cell proliferation. Biallelic loss of NF2 is required for tumorigenesis and occurs via mutations, loss of chromosome 22, mitotic recombination, and DNA hypermethylation. Thank you for listening to this lecture. I hope that this overview has provided you with a sense of both the genetic alterations that underlie pediatric brain tumors, as well as how these genetic alterations might be exploited to provide hope for new targeted therapies. This slide contains a list of references from which images are obtained for this presentation, and a complete list of references available with the online version of the text. Thank you for your time.